Yes, ma'am, it's visible. It's visible today.
Okay, okay. I'll start the lecture, okay?
Yes, ma'am, you start.
Mhm. So last time we have seen the pathogenesis of ischemic and hypotoxicity injury. So I'll just give you an overview. Okay, what are the things we need to study in this chapter, and today what are we going to cover? So in this, we are going to see the pathogenesis of everything. First is ischemia, the physical component, the chemical. Okay, the first is this ischemia and hypoxity. In that, we have two things: reversible cell injuries, which we can recover, and the other is irreversible. Okay, and these are the different components where it goes. So this irreversible. Then we are going to see this is the part of irreversible. Okay, Then we'll see the stress protein in cell injury, the second part. The third is pathogenesis of the chemical injuries, pathogenesis of the physical injuries. Okay, these are the three things we have to cover when it comes to the pathogenesis of the cell injuries. First is the physical, chemical, and the other is toxicity of oxygen. Okay, so last time I have explained this diagram. The same thing happens in reversible and irreversible. We'll just go in deep. We'll just study in deep what is happening at the membrane of phospholipid, what is happening with the toxicity of calcium, and what happens when we have the oxygen radicals, toxic. In deep, what happens in cell membrane, the influx of calcium and sodium at the cellular level. I've explained you this, but we'll see in deep now. So the first is reversible cell injuries. If the ischemia and hypoxia is of short duration, the effect may be reversible on rapid restoration of the circulation. Example, in coronary artery occlusion, myocardial contractility, metabolism, and ultrastructures are reversed if circulation is quickly restored. Okay. So first in this reversible, we are going to see all the examples where the cell recovered itself and how it does and to what extent the injury is happening. Okay, and to what extent the injury can happen and we can reverse. These are the factors we are going to see under this. The first, we are going to see the decreased amount of generation of ATP. Okay, the first part, as you all remember, in ischemia, hypoxia, we have seen reduced ATP generation. Then there was the toxicity of the calcium in mitochondria, cytosolic. Oh, sorry, just a minute.
Ma'am, we cannot hear you. You are speaking, but we cannot hear you.
Okay. Okay, am I audible now?
Yeah, ma'am. It's audible now.
It's comfortable.
Yeah,
Okay.
it's
So what I was saying, as I explained you last time, ischemia and hypoxia, what is happening in that is the ATP is getting involved, calcium is involved, the membrane is involved. At the end, what is happening? The membrane damage is happening. So you need to remember that. Okay, what are the things we are going to see in cell injury right now in reversible and in irreversible is the ultimate of membrane damage. Okay, something to that extent that we can recover, and then we'll see what are the things happen where from where we cannot recover the cell injuries. It undergoes in cell death. Okay, the first is decreased generation of cellular ATP. So first we are going to see what are the things we can recover, so the reversible injuries. So in that, what is happening? Okay, damage by ischemia from the interruption of versus hypoxia from other causes. Okay, all living cell requires continuous supply of oxygen to produce ATP, which is essentially required for the variety of cellular functions. Okay, ATP is human cell derived from two resources. So what are the source of ATP? First is the oxygen source, the aerobic source that we get. Then the other is anaerobic. We all know that ATP is generated with the carb cycle as well, and from anaerobic as well. When we produce lactic acid, along with that, we also have ATP. Okay. So what happened when we are having reversible cell injury? The first thing happens is decreased generation of this ATP. So ischemia, due to the interruption of the blood supply as well as hypoxia from other cause limit the supply of oxygen to the cell that cause decreased ATP generation. So you need to understand one thing when it comes to reversible, we are taking one component and explaining. They have explained one component. So the first ATP generation has been explained. Okay, what is happening when we don't have oxygen, ATP generation won't happen. Okay, is it clear till here? Any
yes.
confusion? We won't go to the end. We're just taking one components and we are understanding what is happening at that level. When it comes to calcium, then we'll see what is the exchange happening between calcium and sodium. Okay, those are the things. So just remember the components which I have explained last time, na? The ATP, the calcium, the lipid breakdown, protein breakdown, and the loss of membrane. Okay, I'll keep on reminding you, but just have a hold on that. So these are the changes that are happening. You don't have to remember this diagram, but it's for information. You have to see first, this is mitochondria, the healthy normal cell. What happens? The swelling happens in mitochondria, which is, if it is happening swelling, then it is reversible cell. What happen if the swollen molecules and large density, if it's happening at the larger level, we cannot reverse it. So you need to remember, okay, what are the things happening in cellular level. At mitochondria, the swelling happens. In membrane, what happens? There is a cell swelling, myelin figures which are present in that, they break down. The membrane breaks. This is a membrane, na? It breaks and become like this. There's a disruption of the membrane that happens. And the same at the ribosome, the outer layer of the ribosomes doesn't, they start disappearing in ribosome. These are not the something you need to remember, and we do not even see those detail in any reports, but just for knowledge, if you remember, or if you know, have a thing just to know. At lysosome, what happened? Autophagy means what happened, this really gets bigger in size compared to other cells in it. Okay, they again got swollen so much that at the end it rupture. They cannot do the proper function of their. Now, we were seeing the ischemia part. There's the second point in this is, in ischemia from interruption of the blood supply, aerobic respiration as well as glucose availability are both compromised, resulting in more severe and faster effect of cell injury. Ischemia cell injury can cause accumulation of metabolic waste products in cell. So what are the metabolic waste products? So what happen when we do not have oxygen, proper oxygen in our cells, they cannot carry the waste. Okay, who are carrying the waste of, when we have oxygen, the cell take this oxygen and move. They flow through our veins and arteries, carry the waste product and deposit to the kidney or deposit to the liver, and those are the things happening. But when we don't have oxygen, when RBCs are not at the optimal level, so these waste products are going to stay in the cell itself. Those are the lactic acid or some high level of sodium, high level of calcium. Okay, so these are the things stays in the cell, and at the end of the cell cannot perform its function. So when we don't have oxygen, these are the things happen. Other is, on the other hand, the hypoxia from other cause. What are the other cause? RBC disorders, anemia, or those are the things, thalassemia, heart disease, lung disease, anaerobic glycolytic ATP generation continues, and this thus cell injury is less severe. What are they saying in this is, when we have two system which are working for our cell, first is aerobic and the other is anaerobic. If we have disruption in our aerobic system, to some extent, we cannot say 100% anaerobic is going to take care of it, but to some extent anaerobic will work more and take care of those things. But the one drawback we have in anaerobic cycle is, there is a large amount of production of lactic acid. Now, one example I'll give you. When you run for 10 kilometers or something, you're having, why do we get domes? Why do we have pain in our calves? What is happening? Can anyone know? Does anyone know about it? When you run for 2 minutes or 3 minutes, you get tired, you feel some cramp in your calf. What is happening? Anyone? So what happened that our oxygen, what happened when you start running, when you start doing any activity, just imagine to the exertion level. Okay. So you started running, either you have to run for 1 kilometer. Okay. Then you start running. What is happening at cellular level? Your ATP, organic aerobic cycle is start working. but after few minutes it gets over. Then your anaerobic is working. It starts accumulating the lactic acid in your cell, and that is the reason you get tired and you cannot run more. Okay, in marathons, in athletes, what we do, we keep on training them and we increase the lactic acid tolerance in them. Okay, so these are the waste products that comes into it. So what are they saying? In the other hand, even though there is a hypoxia, aerobic is not working at all. Anaerobic will take care of to some extent, but at what cost? It will accumulate the waste products over there in the cell. It is reversible, but at this cost. Okay, is it clear to everyone?
Please, madam, screen share.
What? What are you saying?
जी जी मैम.
Is it clear till now?
Clear.
Okay. So now the second point is, first we have just seen the hypoxia, what is happening. First point was of ATP. Now the second is at the mitochondrial level, we'll see what is happening. So in intracellular lactic acidosis is happening. Now we know that aerobic has been done. Now the second is anaerobic. There we have lactic acid accumulation. So that is the term we use is lactic acidosis, when there is too much of accumulation of lactic acid. So now due to low oxygen supply to the cell, aerobic respiration by mitochondria fails first. This is followed by the switch of the anaerobic. That is glycolytic pathway and requirement of the energy. This results in rapid depletion of the glycogen and accumulation of what? Lactic acid, lowering the intracellular pH. Early fall in the intracellular pH result in clumping of nuclear chromatin. Now, from this, what you people can understand? I have explained this, the lactic acid. Hello, do you want to say something? See, this might be a new thing for you. So it's okay, we can go slow because these are very basic things that you need to know when it comes to cell, how it's work. If first oxygen, from where we are getting oxygen, how we are using that oxygen at ATP levels and that. Till here it's clear? Now the third point is damage. First we have seen the ATP, that is oxygen. Second we saw the anaerobic one. The now third we are seeing the damage of the plasma membrane. How it is happening with the swelling of the plasma membrane because of the accumulation of the waste products or other things, calcium or sodium. Okay, the lack of ATP interferes in generation of the phospholipid from the cellular fatty acid, which are required for continuous repair of membrane. Okay, this results in damage to the membrane. Pumps operating the regulatory sodium, potassium, and cell calcium are under. So what is happening? If we need oxygen to repair that membrane, but as we do not have that, so what is happening at the membrane level? It cannot get repaired and it is getting breaked, as we have seen over here. This one, cell swelling, myelin figure, and it get breaks, rupture. Do you have any doubt? Anyone? Should I continue? So what is, okay, I'll make it more simple to that. Just forget about the membrane. Now, first we have seen the aerobic. Second, we saw anaerobic. Okay. Now, third is at the membrane level. What is the function of the membrane? What happened? We have sodium, we have potassium. So we take out the sodium, we restore the potassium in our cell for the normal function. You just need to know how many sodium, how many potassium, do not go into that. Just understand one thing, you have one cell, there is a cell membrane to it, and we have sodium and potassium. Now the influx, mean the movement of sodium out and potassium inside the cell is required for the normal function of cell. Now what will happen that there is a cell injury. So with the reduced amount of oxygen and with the reduced amount of ATP, this movement is going to get compromised. Is this clear to all? Can
Yes,
anyone reply?
clear.
Clear na? Huh. So I'll just read it out, just understand. कि failure of sodium potassium pump normally, the energy ATP dependent sodium pump. We need energy to do everything. So when we do not have ATP, now what is happening at cellular level? The sodium and potassium pump is getting compromised. So normally what happens? The transport of the sodium out of the cell and the diffusion means coming in, the diffusion of the potassium into the cell. This is the normal function. Now, with the lowered ATP in the cell lowers the activity of the sodium pump and consequently interfere with this membrane regulation. So what happened? The potassium, the sodium and the diffusion of potassium out of the cell is reduced. Now sodium is not going out of the cell. Where is it? Is into the cell now. Okay. So the accumulation of the sodium in the cell leads to increased intracellular water to maintain isosomatic condition. क्या हो जाएगा? Swelling. The cell will get swollen. Why? The sodium is not going outside, not allowing the potassium to come in because we do not have ATP for that. Okay. So it's clear for you that we do not have oxygen, so ATP is not going to get produced, so the organic way is compromised. Then we took the anaerobic part, even that we had a drawback of having waste products. Then third, what is happening at a membrane level, sodium and potassium, these three things have been compromised. This all things are reversible. Okay, you need to understand, but I'm just showing you the function. Now the fourth part is reduce protein synthesis. That protein synthesis happen at ribosome in our cell. We have talked about the mitochondria, we have talked about the cell membrane, now we are talking about the protein. So what happened when we do not have oxygen, everything is getting swollen up. Okay, so even ribosomes get swollen up. We have seen that in our picture. This gets swelling and the outer layer get disappeared. Okay, so that is going to happen over here. So as a result of continued hypoxia, membrane of endoplasmic reticulum and Golgi apparatus swollen up. Ribosomes are detached from the granular. This would, I'll just make it simple for you. Just give me a minute. So what they are saying, ribosomes get detached from the granular rough endoplasmic reticulum, and okay, just do not go into this depth. It will be very difficult and very cellular. Just remember one thing. What is happening? The protein synthesis is getting reduced because the ribosome is getting swollen. Why I'm saying this? Because we have seen here protease activation is reduced here because yes, and any doubt? Can anyone just randomly just tell me the 4 points that I included today in the reversible? Did you understand, or you anyone can just type it out? I'll read it out or something. That it was clear. This part, ATP part was clear. The second is anaerobic part. Third was plasma membrane, and the fourth is the protein synthesis that get reduced. These four are clear to you all. When the injuries are to this level in the cell, the ribosome, the cell membrane, we can do it reversible. Because in after that, I'll go irreversible. See, irreversible, everything is happening to the overextend. Everything is happening the same way, but in the larger amount. Means we do not get the oxygen for longer period of time. So what will happen? It will get swollen and it will go into the rupture. Okay, the ribosome is getting swollen up, and in irreversible, what will happen? It will rupture. Okay, the cell membrane is also getting ruptured. So everything, the same functions are going to happen, but to that extent that we cannot reverse it. So it's very simple for you. That persistent ischemia or hypoxia result in irreversible damage to the structure and function of the cell. Now there are two essential phenomena that distinguish irreversible and reversible cellular injury is what first is inability of the cell to reverse the mitochondrial dysfunction on the reperfusion or reoxygenation. Now, what they are trying to say here is just imagine if we have stopped the oxygen for 2 minutes. Okay, in one place, we in one cell we have two cells. In one cell, I have stopped the oxygen for 1 minute, and in other, I have stopped for 5 minutes. Then, what I do after 1 minute, I give the larger amount of oxygen. I start the supply of oxygen. What will happen? Everything will start recovering. The mitochondria, the ribosome, the cellular membrane, the sodium calcium, sodium potassium influx will start happening. Okay, but under other condition, after 5 minutes, if even though I'm giving the oxygen to that cell, the functions are done, we cannot reoccur, it cannot reoccur, and everything will not go to the normal. Okay, the swollen thing, the rupture thing is not going to get to back to the normal. The sodium potassium pump will not start even though we are giving the oxygen. So to that extent, we say it's irreversible. When we again give the oxygen to that injured cell, but it cannot recover. Is it clear now?
Yes, clear, ma'am.
Okay. So the second thing is again the disturbance in the cellular membrane function in general and in plasma membrane. So what happened? In addition, there is a further reduction in ATP, continuous depletion of protein, reduced intracellular pH, leakage and everything. The biochemical changes have affected to the ultrastructure of the components of the cell that we have seen in 2.3b, this one. Okay. Now they have just taken few points as we have seen in reversible. Na? So to what extent irreversible, what are the components are going to get involved in irreversible? The first is calcium. As a result of continued hypoxia, a large cytosolic influx of calcium ion occurs, especially after reperfusion and irreversibly injured cell. Excess intracellular calcium collects into mitochondria, disabling its function. Morphological changes are in a form of vacuum. See what they are saying is when there is no oxygen, na, as we have seen in this diagram, when we do not have the oxygen, what was happening? The amount of calcium was getting accumulated into the cytosolic. So it interrupts the function of the mitochondria and cannot reverse the cell injury. Okay. So what is the function of calcium? What is the role of calcium in irreversible? It gets accumulated in the mitochondria and do not allow the normal function to happen over there. Clear? Now the other thing is activated phospholipase membrane damage. Now, this is a very important function that is happening in irreversible cell injury, because what happened? There's an increased cytosolic influx of calcium in the cell, activate endogenous phospholipase. Phospholipase is nothing but the fat we have, na? Lipids we have, that is getting activated, and in turn degrade membrane phospholipid progressively, which are main constituent of the lipid bilayer membrane. Beside, there is also decrease in replacement synthesis of the membrane. There are two things happening. There is a decrease in phospholipid and there is a decrease in production or replacement of the membrane because of the reduced ATP. How the membranes are getting damaged because of the phospholipase that has been explained over here. See this phospholipase activation because of the increase in calcium. So we have the calcium over here, then it's increase in the mitochondria. What is the next step is happening is phospholipase activation is happening. So that will reduce the synthesis of the cell membrane. So now there won't be a reversible thing. Now the membrane is getting damaged at this level. So you have to see the loss of phospholipid is also happening. Lipid breakdown is happening, and that will ultimately break the membrane damage. Understood? If you do not write, you do not have to explain each and everything. You can just make the diagram, but for your understanding, I'm explaining you. Okay. In exam, you do not have to write this many things. You can just give a heading or you can just make that diagram. But the diagram is important. Now, the third thing is intracellular protease. Now, what is happening at cytoskeleton damage? The normal cytoskeleton of the cell is microfilament, microtubules, and intermediate filaments, which anchors the cell membrane, is damaged due to the degradation of activation of intracellular protease or by physical effect of cell swelling, producing irreversible cell membrane injury. So, normal what is happening? The cell cytoskeleton damage means the layer of the cell is getting damaged because of the swollen up. There is no space, there is no normal thing happening, there is a accumulation of certain things. So the damage is happening to that level where it is irreversible. Now the nuclear is also getting damage to this, like piconosis, cryolysis, and cryolysis. The nucleus get damaged, the DNA get damaged in this. The other thing is lysosomal damage, that phagocytes. In this, what happened? The membrane has the lysosome that is getting damage in that through the enzymes. Okay, you do not have to go into that LDH, SGOT, and everything in deep, but it has been explained over here. See this one, serum enzyme estimation because of the lipid peroxidation DNA damage. Reversible, irreversible, clear to everyone?
Okay, madam.
See, you do not have to remember all this in detail, but if someone asks you कि cell damage में होता क्या, what is happening in cell damage, so you should be able to say कि there are two process, to some extent reversible, and if these are the things are happening, like lysosome is getting damaged or there is increased lipid peroxidization is happening, so it's irreversible. Irreversible, what is happening? If the oxygen has been interrupted for few seconds or few minutes, you can reverse it, even though with the accumulation of the lactic acid. So these are the things you should know when the injury is happening. Okay, this is the basic thing. But in exam, if I ask you, you just have to make this diagram. Understood? Tomorrow I'll take this one. These are not important right now. These are the same thing, radicals. We won't take that. We'll take this one. Pathogenesis of the chemical injury and pathogenesis of physical injuries. Okay. Is it clear now?
Clear, ma'am.
Any doubt?
No.
Okay. So Monday we are having lecture, right? Said, we have lecture on Monday, right? Said is not there. Okay, Monday we have lecture. So we'll just carry out that physical and chemical part. Okay.
Okay, ma'am.
Okay. Thank you. Anyone wants to ask anything or any doubt regarding this? No?
Please say again example.
Which example? Of the cell injury? See, in irreversible, if there is a fungal infection happening, in irreversible, when there is a necrosis is happening, what happen in necrosis? There is no oxygen over there. Gangrene is happening. Those are the cell injuries which are irreversible. But one is having a normal cut, one is having a normal burn or a boil, that is reversible. After some times, what will happen? The oxygen will go there and it will get recovered. But in necrosis, in fungal infection, these are the things, there is a blockage or there is a damage, continuous damage to the cell membrane. In infection, continuous blockage of the oxygen in necrosis is happening. Okay. So there you can see irreversible cell injuries. Clear?
Ji, ma'am.
Okay.
Okay.
Mhm. Thank you.